So just a reminder, first exam is a week from Tuesday. So I sent out a study guide like 5 minutes ago. Yeah, 5 minutes ago, just before I came up here. And I also attached an extra credit article. So did I mention that the exam will have, usually about 3 questions pertaining to the article. It's a 100-point exam. I think usually about 6 points worth of extra credit. Again, if you want extra credit, learn something extra. That's the way I look at it. All right. So it's up to you if you want to read it and be familiar with it. The questions should only really be answerable if you read it. That's the idea. Okay. So again, it's extra credit, so it's optional. So it's up to you. All right, and the exam. I also sent you a notice. Jeff, who you probably saw when we introduced you to the Science Success Center down the hall, he's going to host a review session for this class on next Thursday. Yeah, a week from today. And I think it's from 2 to 4. But I put it in my note. And it's going to be actually in this room. So if you're available at that time, show up. Okay. If not, then you can try my office hours. Okay. And always, if you really want a little help with anything, and their availability doesn't work, my office hours don't work, just talk to me. We can arrange a time. Okay? I can stick around after lab or something if you need to. Excuse me. All right, so any questions before we address chapter 11? Now, this is kind of let's get acquainted with the world of bacteria. Okay? We've been looking at some general features of bacteria, but now we're going to look at some specific ones. So we're going to see some major groupings of bacteria and look at some, you never know what's going to be called, some specific groupings and some examples of some species in each group and their significance. Okay? It may seem like, it's one of those chapters where it's just kind of, conceptually it's perfectly understandable. It's just kind of stuff to know. Okay? And so, people always ask, do I have to know everything on there? Well, what I usually say is, as you'll see when we go over and look at some examples of members of different groups, just be familiar with, know a couple of organisms that fit into a given group and what the general characteristics are of that group. That's the idea. And on the exam, you might have, you'll have maybe a multiple choice question or some matching or something to help kind of see if you remember some of that. All right? So let's start with the bacteria. The archaea, we're going to just spend just a couple slides on that, just to say a few things about them. By the way, I'll say one thing right now. The archaea are not nearly as well understood and characterized as the bacteria. And there's a couple of reasons. The bacteria are a lot more relevant to us for good and bad reasons. The disease-causing, prokaryotes, bacteria. To my knowledge, there's still no archaea that actually causes human disease. And as far as, living on our bodies, in our guts, in our skin, doing good things in the soil and so forth, pretty much, for the most part, it's all the bacteria. The archaea are really fascinating, but they're very difficult to study. They're hard to grow. So we kind of know how to grow a lot of these. How to grow a lot of these is very difficult. So, but gradually they're getting more successful in analyzing them and so forth. So the breadth of knowledge about these guys is expanding, but for now it's fairly limited. So we're going to focus on the bacteria. Did you see we actually have 3 informal groupings. When I say that, you know what I mean? These are not taxa. Okay, we're not talking about a kingdom, a phylum, a class, or anything like that. They're just sort of a convenient, unofficial grouping. Okay, we do have, phyla and classes and so forth that we'll point out as we go along. Phototrophic. Okay, these are the photosynthetic bacteria. And we have the, of course, gram-positive and gram-negative. You should be familiar with. Okay, so before we start looking at some of these groups and what their characteristics are and a few relevant examples, we need to get familiar with a little bit of terminology. Some of this should already be familiar to you and some may not. Aerobic versus anaerobic. I'm sure you've heard that before. And, so again, aerobic organisms either require, like us, or are simply capable of existing around and using oxygen as part of their metabolism. Okay? Anaerobic organisms, they don't need oxygen, or they can simply do without it. Okay? Or some of them actually cannot even tolerate it. So I think I mentioned before, some organisms, oxygen's a poison. And we'll address why in I think it's chapter 6. But oxygen and metabolism involving oxygen tends to produce, you ever heard of reactive oxygen species? Or you could even call them oxidants, because we have antioxidants to deal with these derivatives of oxygen that can be actually very damaging. So we're prepared, all aerobes are prepared to deal with that. But your anaerobes that can't tolerate oxygen, the byproducts that can be harmful, they're not prepared to deal with them. So that's why they can't tolerate it. Okay, so aerobic versus anaerobic. Probably should already know that one. When we're talking about the use of energy, okay? So where does the energy come from? So the organism has to acquire energy from some source. Energy that's going to ultimately end up in ATP, which will then fuel all kinds of cellular activities. So phototrophs, their ultimate source of energy is light. Okay, photo, of course, you probably know means light. So phototrophs, literally, the name troph refers to eating or feeding. So they eat light, which, sounds silly. They don't eat light bulbs or anything like that, but obviously they acquire energy directly from light, okay, by photosynthesis. As we'll see, photosynthesis can happen in more than one way. So phototrophs, and then we have chemotrophs. Okay, chemotrophs, guess where they get their energy from? They get it from chemical forms. Okay, those chemical forms can be organic molecules, which we can say organotrophs, organic eaters, or lithotrophs. Lith like otolith, or I don't know. That's the only one that comes to mind. The little stones in your inner ear. Lith means stone, okay, or rock. So they're rock eaters. Basically, that just means they're getting their energy from inorganic sources, inorganic chemicals like iron or something like that. Okay, these aren't really all that common. These are much more common. Okay, so if you were to go describe yourself to somebody, you would brag about being a what? Are you a lithotroph? No. It would be very good for your teeth. What's that? You could say I'm a chemotroph, and then if anybody knows, they would say, well, what kind of chemotroph are you? Of course, you're an organotroph, yeah. Okay. Exciting stuff, isn't it? All right, so we also have some other prefixes or terminology that are relevant to not only where we get energy, or organisms get energy, but also where they get carbon. You got to have carbon. You need carbon to build organic molecules. Remember, what's an organic molecule? I don't know if we addressed that, but an organic molecule is you sell them at Whole Foods, right? They cost more. No. Organic molecules have, go ahead. Yeah, carbon and hydrogen, and specifically carbon-hydrogen bonds. Yeah, so they're hydrocarbon-based molecules. And for the most part, not entirely, but for the most part, hydrocarbon-based molecules, carbon and hydrogen-containing molecules, are produced by living processes. Hence the word organic. All right, so what else we got? So carbon, okay? So where do you get your carbon from, okay? We eat it, so we are heterotrophs. Why are we called heterotrophs? Because our carbon comes from basically molecules, organic molecules produced by other organisms. So not only do we get energy, from organic molecules, we also get carbon we need to build our own organic molecules. So probably most organisms out there are heterotrophs. Autotrophs, on the other hand, are self-feeders. Auto meaning self. And anything we call an autotroph, it can get its carbon from CO2. Okay, CO2 just out there. So, so plants, for example, most photosynthetic organisms, their carbon just comes from carbon dioxide, and they carry out a process you may have heard of, carbon fixation or the Calvin cycle, to using NADPH and ATP to produce organic molecules like glucose. All right, so you can put a couple of things in front of troph to describe an organism. So you can see a chemoautotroph or even an organoautotroph or whatever you like indicates energy source and source of carbon. And a couple other terms we will use quite a lot, okay, are facultative versus obligate, also sometimes called strict. Okay? So every single one of you is an obligate aerobe, okay? Also a strict aerobe. You have to, you require oxygen, okay, because you can't live without it, at least not very long. If you happen to be a yeast, like brewer's yeast, you're facultative, okay? It means you're capable of going with or without, for example. So a facultative, very common term is a facultative anaerobe, okay? It has the ability, okay, or the faculty, if you want to say, of being able to survive using oxygen or going without it. Okay? And these terms are generally used to refer to facultative means able to do the less desirable thing, but can do it if necessary. Okay, so if going without oxygen is actually less efficient in terms of energy, because oxygen can be used for respiration, as we'll see in chapter 5. You can make a lot more ATP, per molecule than you can, for example, by fermenting, which doesn't involve oxygen. But some organisms, they can get by just doing that, even though it's less efficient. If the oxygen is not available, if humans lock the yeast in a big chamber with some grapes or hops or whatever with no oxygen, they have no choice to survive but to ferment. They can do it, we can't. All right, so for example, facultative aerobe is not a term that's usually used. Facultative anaerobe means capable of being an anaerobe, but will be an aerobic if that's an option. All right, any question on these terms? Yes, bless you. All right, let's start with the bacteria, and we'll start with the bacteria that are photosynthetic, the phototrophic bacteria. Actually, photoautotrophs, technically. All right, and we're going to look at a couple of examples. Okay, and again, this isn't really this isn't an official taxa or taxon. Okay, it's just a descriptive term for bacteria that are capable of photosynthesis. And I'm sorry, I'm getting ahead of myself here. This is just these are just a listing of the groups we're going to look at. We've got some phyla in each category, so don't worry about this. Just kind of a little list of what we're going to look at. So let's go look at the phototrophic bacteria. Okay. So we're going to start with the cyanobacteria. Probably heard of them. They used to be called the blue-green algae, but we don't call them that anymore. Any idea why? Because they're bacteria. What are algae? Algae are protozoans. They're eukaryotes. Yeah. So these are not even remotely algae. So, excuse me. Here's one example called Anabina. So this is the genus Anabina. Here's a picture of it. You may have seen these in some of your other biology classes. Don't worry about the details. Here's another example. The key they're very, very important ecologically. Okay? Any autotroph is going to be the base of the food chain or the food web. Okay? Because all the other, heterotrophs depend on those organisms producing the organic molecules that gradually work their way through the food chain as a source of energy and carbon. So, so these are all very, very important. So not only is producing organic molecules by photosynthesis important, it's also the production of oxygen gas. As we'll see, there's photosynthesis that produces oxygen and there's photosynthesis that does not. We'll see examples of both. So these are oxygenic photoautotrophs. Okay? They produce O2. Where does O2 come from? We'll put that up there. But O2 in photosynthesis, the light reactions, O2, the oxygen in O2 comes from one of our favorite molecules. Water, yeah, H2O. Okay? So literally the hydrogens, using the energy from light, it results in water being split up into hydrogens and oxygens. And the oxygens basically are a, quote, waste product, even though it's actually useful, obviously. But from two water molecules, the oxygens combine, and that's the byproduct of oxygenic photosynthesis. And that's why we have an atmosphere that's almost 20% oxygen, thanks to organisms like these. Okay? Something else I always like to point out, good old nitrogen fixation. Why is, so basically converting nitrogen in gaseous form into nitrogen in accessible forms to plants and other organisms. So it gets into the food chain. Remember, you need nitrogen. Why? Because there's a lot of, for amino acids and proteins and the nucleotides and nucleic acids, DNA and RNA, and many other molecules require nitrogen. So nitrogen has to be accessible to an organism. Whether you eat it, like us, whether you get it from nitrates or ammonium ions in the soil for plants, it has to be in those forms. And it really goes from nitrogen gas to these ammonium nitrates in the soil into plants. And from there, the nitrogen gets into the food chain, and ultimately we can get some. So this is a hugely important process. Most organisms can't do this, but a number of these can, in particular these cyanobacteria. So they're actually triply important people, right? They produce oxygen, they produce organic molecules to get carbon into the food chain, and they also fix nitrogen to get nitrogen into the food chain. Now, we also have photosynthetic organisms that they carry out photosynthesis slightly differently. And as a result, they're not so relevant to us anyways. They are anoxygenic. Let me just get them all up here. So they're anoxygenic. Obviously, that means they do not produce oxygen as a byproduct. That's very simply because they get their hydrogens they need for photosynthesis from a different molecule, not from water, but from something else. And for the sulfur bacteria, it's from hydrogen sulfide. Okay? So it should not be your favorite molecule. It's poisonous. We don't want to be on that. And byproduct is going to be S, it's going to be sulfur, okay, as opposed to oxygen. We also have some organisms that are photosynthetic, and again, they're not using water, they're also not using H2S, they use organic molecules as a source of the hydrogens. And again, the light energy is being used to split molecules, separate the hydrogens from the rest of the molecule, and the electrons from the hydrogens are what are used in the light reactions to generate the two molecules I asked you about on the last quiz were those. So the light actions of photosynthesis, they make DP and NADPH, okay? So it requires electrons in an energized state. The energy comes from light. Those electrons can come from hydrogen in water for some organisms, or from hydrogen sulfide, or even organic molecules in some cases. Now both of these, you can see they're obligately aerobic. Okay? So they're examples of organisms that cannot survive in the presence of oxygen. So you don't find these out there. They're not abundant in the oceans, or that they tend to be in kind of stagnant, boggy environments down below where there's really not much oxygen. So they tend to also, the sulfur ones, they tend to have inclusions. Remember inclusions? They're sort of, I guess, to a vacuole in a eukaryote, but the difference is the aggregate of materials sort of being stored over inside the cell. It doesn't have any membrane around it, so it's not a vacuole. It's just a little cluster of some material we call an inclusion. Okay, so we've got oxygenic as well as oxygenic bacteria, all of which we call phototrophs. Now we're going to look at some things that seem perhaps a little more familiar. We're going to look first at gram-positive bacteria. I wish it would stop doing that. I don't know how to make it stop. Can you still see okay in the back? There we go. So again, this is an informal grouping, but within this are some formal taxa we're going to look at. Two phyla, kingdom, phyla, order, etc. The Firmicutes and the Actinobacteria. There's going to be some interesting organisms in here. I always point out good ones and some bad ones. You're probably interested in the ones that cause disease. But we like it where there's some good done out there too. Firmicutes and the Actinobacteria. You can see we have some pathogens actually in both.
And a sort of crude way to distinguish from each group is based on DNA sequence in terms, basically GC content. So what does that refer to, G plus C content? It's the percent. So what's E's and C's in DNA? What are we talking about? Right, guanine and cytosine. So in DNA you have these integral base pairs, right? And there's only two base pairs, even though each can pair with only one partner. Remember what those are? So A, adenine can only base pair with T, thymine, and vice versa. There's AT base pairs, depending on the side of the base pair you're thinking of. There's just AT and guanine cytosine, okay, GC base. So what centome is GC base pairs for AT base can be very useful for just kind of putting out or sort of determining if it's say a known bacteria part of a group or another. You have to do more tests to see exactly what it is. But for example, a gram-positive bacterium and content high greater than 50%, well, it's in this group. It's not going to be a group. So, you have a we'll see that. Look, a bacteria is GC content over. So, let's start. We have genus, plural, crap. Genera up here to zero. Disease causing gene base. But it's so multiple strep staph, dermis, apha, so whatever, so much age, much example, one actually some of this. So, yeah. So I guess, you know, if you're interested in weaponizing bad organisms, I would suggest don't ever do that, by the way, just for the record. But endospores would be a good choice. Something that produces endospores would, you know, through the packaging and delivery process could remain intact and capable of infection. Because endospores, you know, again, they can survive the harshest treatment. And then what happens if you happen to breathe in endospores that are in the egg? They suddenly,
Come back to life in your body.
yeah, exactly. They suddenly find themselves in a warm, moist environment, lots of nutrients, and as they say, they'll hatch and develop back into vegetative cells, divide, it can cause infection. Okay, so Bacillus anthracis. And then we have these, I'm sure you're very familiar with these diseases. Clostridium is the genus that has a lot of, you know, very dangerous pathogenic bacteria. These are just two. And by the way, it's not here, but they're actually obligate anaerobes. So all species of Clostridium are obligate anaerobes. So Clostridium tetani. So for them, being in contact with the air means I need to become an endospore, I'm going to die. Okay? So where do you get tetanus from? Just, yeah, you think like the rusty nail thing or any kind of puncture wound, especially something that's been in contact with the soil, because any animal that may have passed some of that material, endospores, into the soil, it's just going to sit there and gradually, you know, mix in, and if anything comes in contact with it that punctures skin or something, endospores can get in there and cause tetanus, which, you know, we can treat that now, but that can be a very unpleasant and potentially deadly disease. And certainly botulism as well. So botulism, again, an obligate anaerobe. What's the main risk of contracting botulism? You ever like people who, was that?
It's like a toxin.
Well, yeah, there's a toxin, exactly. So this bacterium causes its disease, botulism, by producing a toxin. And, you know, you heard, you know, people who have, like, in the past, whole families have died because they all ate some kind of home prepared and canned or packaged food material. And if it's something that's eaten without heating, what happens is in the sealed container, it becomes an anaerobic environment that the endospore can then hatch and grow and produce the toxin, and then people eat the food, and it inhibits, you know, the neuromuscular junctions, and people can't breathe and they suffocate and die. That's botulism. The toxin can be inactivated by heating, but if it's not heated sufficiently or heated at all, that's when these things happen. So yeah, so home preserves and things like that got to be really, really careful. Okay, there's a few others too. You've probably heard of C. diff, especially those of you who work in the hospital environment. Clostridium difficile can cause really, really dangerous, you know, ulcerative colitis that can kill people. And by the way, the large intestine, a nice, warm, anaerobic environment for a lot of things. So, in particular, that species. Okay, let's look at a few more to make mention of. Okay, here's one that's not bad. Lactobacillus, so that's a genus, and this can be found, you know, on your skin or other places. It's part of the normal, natural, healthy microbiota. Female reproductive tract, also used in, you know, fermented foods and so forth. So that's a good one. Mycoplasma, these are of note. They're kind of interesting. These are extremely small bacteria. They're obligate intracellular pathogens. Okay, so what does that mean? They can only survive and thrive and reproduce inside host cells. They're also interesting in that, unlike other firmicutes, they're gram-negative, and they don't have a cell wall. Okay, now as we know, a gram stain, you got to have a certain kind of cell wall to trap the crystal violet iodine complex. Remember that? So these guys don't. They can't trap it because they don't have a cell wall. Yet they're in this phylum. So superficially, they seem like they don't belong here. Any thoughts as to why they're included in this group? We're in the age of molecular biology where we have a lot more information than just what things look like and what kind of, characteristics, physical characteristics they have.
Isn't the glycan something that makes them?
Oh, that's a good guess, but they actually don't. So there's no peptidoglycan, there's no teichoic acids. So they're completely, in terms of cell wall, they have nothing in common with other firmicutes.
So it's not the outside structure?
No, it has nothing to do with it structurally. What I'm getting at is really a lot of classification nowadays comes down to DNA sequence homology or similarity, right? And so even though things superficially seem very different, when it gets down to it, if they got like 98% sequence similarity and something else is 80% similar, well, you know what? They've diverted evolutionarily at different times. The ones that are more similar have a more recent common ancestor and really do have more in common with each other, even though superficially they may seem different. So these guys are going to have a lot more DNA sequence similarity, genetic similarity with other members of the phylum Firmicutes than organisms in different phyla. So even though they're missing one structure, it doesn't mean they really don't belong here. All right. So let's move on. And the actinobacteria, so remember these can be crudely distinguished from the Firmicutes because the percent GC in their genomes is different. Okay? It's greater than 50% as opposed to less than 50% with the Firmicutes. I'm going to, again, usually point out at least one good guy and a few bad guys. So there's a good guy. Species of Streptomyces, common soil bacterium or bacteria. And in the soil, bacteria play a lot of important roles, fixing nitrogen, recycling nutrients so plants can take them in and so forth. In fact, if any of you do any gardening in your yard, there are certain fertilizers that they call probiotic, that actually have soil bacteria in them. That should help your plants grow well. So things like this. But there's really, one thing really significant about species of Streptomyces is they're a source of quite a few antibiotics. And we'll learn in chapter 10 that the word antibiotic actually has a very specific meaning. You have things we call antimicrobial drugs. Anything you take, either orally or intravenously or whatever, to kill a microbe is called an antimicrobial drug or antibacterial drug. Anything you call an antibiotic, even though we use the term to refer to all drugs that kill bacteria, for example, anything you call an antibiotic literally means it's made by another microorganism and it has a toxic effect towards others as opposed to itself. So these bacteria make these toxic substances that kill competitors, that kill other bacteria in their environments. And we discovered them and realized, hey, we can package these into pills, purify the substance. They have selective toxicity towards bacteria, but they're not toxic towards ourselves. So anyways, very important antibiotics produced by species of Streptomyces. Diphtheria, a disease you probably all heard of. Corynebacterium is the genus. Corynebacterium diphtheriae is the species that's the cause of diphtheria. Now here's another very significant disease-causing genus that I've already mentioned. Okay, the genus Mycobacterium. Okay, the cause of tuberculosis I mentioned many times. Mycobacterium tuberculosis. Mycobacterium leprae, cause of leprosy. So these are all identifiable. These two by staining. So if anybody has symptoms of either of these diseases, a little sample of material from the patient, whether from the respiratory tract or from the skin or wherever is appropriate, a stain, acid-fast stain will reveal the presence of these guys. So they'll stain acid-fast. They're rod shapes. You see acid-fast staining rods, then that's, again, pretty convincing evidence that that's the pathogen involved. So I said last time I'd address the reason for acid-fast staining today. So now we're at that moment. So the acid-fast staining procedure, what you're going to do next week, and don't write this down. I'm just going to tell you what you're going to do and tell you what's going on with the staining procedure, and then we'll address why these guys retain that primary differential stain, the pink one in this case. So there's a stain, the pink one you start with. It's called carbonyl fuchsin. Don't worry about writing that down, but you're going to use that next week. And generally you expose your smear to it, and you actually heat it. And the heat can be argued as a sort of mordant. It enhances the stain even though it's not a chemical. The heat helps the stain penetrate the cells a little bit better. And then everything, so this kind of sounds familiar. It's just different stains and slightly different procedure, but it's kind of analogous to doing a gram stain, just different stains. So once you do the initial stain with this pink carbonyl fuchsin, everything's pink. So you really haven't accomplished much. You're going to stain everything. Okay? Then you're going to come in this case, instead of iodine to enhance the stain, the heating enhances the stain. Then what's after the mordant in a gram stain, what's next? Right, the decolorizer, very good. So we also have a decolorizer with the acid-fast stain. And the decolorizer is actually acid alcohol. So it's an acidic solution, part alcohol and part, I think it's HCl, to make it an acidic solution. That particular decolorizer will remove the carbonyl fuchsin, the pink stain, from any cell that already is stained with it, okay? With the exception of these, or others like them. Why? Because they have this unusual material in their cell walls. So within their cell wall structure, they also have this stuff. It's called mycolic acid, and it's a sort of waxy lipid.
So it's resistant to.
What's that?
So it's resistant to.
Right, exactly. So this acid alcohol wash, again, assuming you don't overdo it, it fails to decolorize acid-fast bacteria. So in other words, you can think of it this way. The stain is sort of sticking fast to the bacteria, like fastened or whatever, despite the acid alcohol wash. Whereas non-acid-fast bacteria, the acid alcohol wash will wash out the stain and they'll go back to being colorless. Okay? Does that make sense? So the only difference between staining acid-fast and not staining acid-fast is this, hydrophobic, waxy material that helps retain this fairly hydrophobic kind of stain. All right, and just to kind of complete the staining procedure, once you've decolorized and the acid-fast bacteria stay pink and anything else that might be there is no longer colored, no longer pink, what's the last thing you would do in a differential staining procedure like this? You would do a counter stain, right? You do counter stain. And you wouldn't use the one for a gram stain, even though it's a different stain, it's also pink. So that wouldn't allow you to distinguish them. So you use usually methylene blue, for example, or brilliant green, something that's a totally different color. Okay, this is a good time for a break. Let's take a 5-minute break. I do have your quizzes, by the way. I'll get them back to you today. I've also entered them on etudes, so you can always go look at them. So if you're in the Tuesday lab and you want to pick up your quiz, I got it right here. Oh, Thursday, well, because you're going to be in lab anyways, I'll get it to you then. They're going to right after class, so I'll get them first. So, Paul, right? There we go. I want to get a big one in there. I'm sorry, what's it? Isabel. Sancho. Oh, thank you. I just hit the message. I should have mentioned that as well. Okay, and Oh, you're in Thursday lab? Oh, okay. Give me a second, I'll get that for you. I'm just distributing the Tuesday lab. Okay,
I realized I didn't have a problem.
oh, no problem. That's fine, that's fine. I'll check it out for you. Okay, Louise. Oh, you weren't here. Okay. So, maybe the choice is take it as the one you drop or something. Oh, we can just arrange it our own. Okay, I have a new policy this for unexcused pickups. There's a few points. So 13 and 15. I'm trying to discourage, so every semester there's just 2 pickups, so that's fine. Okay. So, actually, I think I have your, today's the last day of my lecture. I'll pass around the sheet to everybody. Okay, don't tell me. Naira? Okay, I'll get yours in a second. Yeah, I'm just getting Tuesdays distributed because they're gonna leave after lecture. But I'll get yours too. Don't worry, I'll get yours too. Ivan, are you here? Ivan Dulce? And Tony Rose? Patsy, are you here?
I didn't do makeup.
You makeup. Patsy Cardinal? Okay, so let's get you Thursday, folks. But you don't want to wait. Ruby? Ruby? Fatima? Daniel? If you don't mind, come on up when I call your name. Tanya? Oh, Ruby. Tanya? Stefka? Angel? And then Brittany? Brittany? And Monica? Jamie? Florence? Callahan. Vic? Do you like to go by Vic? Okay. Joy? Irene? Loretta? Veronica? Caroline? Caroline? Carl? Francisco? Adriana? you are. There you are. And Anastasia. Oh, there you are. Floor. Tam. And Carmen. Oh, sorry. Carmen, there you are. Okay, if you think you didn't get your quiz, it's up here. Oh, I'm sorry. Tanya. Okay, so